Good evening, everyone. I'm Dr. Anoop, a medical oncologist, Regional Cancer Center, Trivandrum. I'll be presenting 4 abstracts. First one is mutational signature analysis reveals patterns of genomic instability linked to resistance to endocrine therapy plus or minus CDK4/6 inhibition in ER-positive, HER2-negative metastatic breast cancer. So the basic study questions in this abstract is, which are the pathological and clinical features of breast cancers enriched for upper BEC or other mutational signatures? Do upper BEC and other mutational processes have an impact on clinical benefit to standard treatment? Which are the genomic alterations that are enriched in breast cancers with upper BEC mutagenesis? So they used clinical sequencing cohort of 4,495 breast cancers, and they have done a clinical genomic analysis and using a machine-learned based detection of mutational signatures from targeted sequencing. And MSK impact targeted gene involved more than 400 genes, and they analyzed somatic mutations and copy number alterations, mutational signatures from non-synonymous and silent somatic single nucleotide variants SNVs by a sigma algorithm in samples with at least 5 SNVs. And what they found out was HR-positive MBCs have higher upper BEC and HRD signatures when compared to triple-negative breast cancers or HER2-positive breast cancers. And breast cancers with upper BEC are linked to lobular phenotype and patient with high mutational burden. Upper BEC and HRD signatures are associated with endocrine resistance, even if the patients on CDK4/6 inhibition plus endocrine treatment. And they have high frequency of specific genetic alteration linked to endocrine resistance like NF1, P10, KMT2C, and IRET1A. And they usually have PIK3C mutations enriched in upper BEC dominant breast cancers. Summary and conclusion: HR-positive, HER2-negative breast cancers, they occur genomic instability during the tumor evolution, and upper BEC and HRD are being the main drivers. So breast cancers with high level of upper BEC are characterized by high tumor burden, and they are usually seen with lobular cancers, and they have specific mutations like PIK3CA mutation, P10, NF1, and CDH1. And upper BEC and HRD dominant HER2 MBCs are shorter PFS in patients starting on first-line endocrine treatment with CDK4/6 inhibition. Coming to the abstract second, clonal evolution and mechanism of acquired resistance to CDK4/6 inhibition in ER wild type and ER mutant breast cancer. Is the acquired resistance to CDK4/6 inhibition due to the pre-existing resistant cells, or is it due to the emerging resistant cells during the treatment evolution? Do the ESR1 mutations impact the clonal dynamics and mechanism of acquired resistance to CDK4/6 inhibition? Or there are any difference in the dynamics and mechanism of resistance to abemaciclib versus palbociclib in ESR1 wild type as well as ESR1 mutant tumors? Here they use the DNA barcoded doxycycle-inducible ESR1 mutant MCF7 cells, and they studied the resistant pattern using DNA barcode sequencing. And what they found out was resistant to palbociclib is likely due to pre-existing clones, and ESR1 mutation leads to selection of different clones. Especially Y537 ESR1 mutation impacts the selection of palbociclib resistant subclones. Coming to the abemaciclib, resistant to abemaciclib is due to the pre-existing clones, but the clustering is strongly driven by the abemaciclib treatment. In abema resistant cells, ESR wild type and ESR mutant cells have high number of overlapping clones. And both palbo and abima resistance may lead to different clonal selection. And difference in the palbo resistant and abima resistant model is more pronounced in the setting of ESR wild type when compared to ESR mutant. And both wild type and mutant ESR1 have distinct clonal dynamics also. In palbo resistant models, ESR1 mutation leads to earlier or more heterogeneous clonal selection, while abima resistant models, ESR1 mutation leads to earlier clonal selection but does not have an impact on the clonal diversity. Interestingly, palbociclib resistant cells retain sensitivity to abemaciclib, whereas abema resistant cells were cross-resistant to palbociclib. And both naive and CDK resistant cells have different transcriptional profiles at single cell levels also. Coming to the conclusion, resistant CDK4/6 inhibition is likely due to the expansion of the pre-existing subclones, suggesting that targeting resistance upfront could delay the acquisition of clinical resistance. Y537S ESR mutation shapes the clonal diversity and dynamics of acquisition of resistance to CDK4/6 inhibition that is more prevalent in palbociclib. Clonal selection and transcriptional changes that occur during the acquisition of resistance to palbociclib and abemaciclib are different, especially if ESR wild type. Coming to the abstract 3, allelic dosage of RB1 drives CDK4/6 inhibition treatment resistance in metastatic HR-positive breast cancer. Here also they use the clinical genomic analysis to identify the mechanism of response and resistance to the therapy. And RB1 loss of function variants are more enriched in JBL and BRCA2 cases. And RB1 and BRCA2 are co-located in the chromosome 13q. And LOH of RB1 and BRCA2 usually co-occurs. And it was also seen that JBL and BRCA2 carries short-term PFS on CDK4/6 inhibition plus endocrine treatment. What they used is that they tried to define the association of pre-treatment RB1 allelic status with the outcome on first-line CDK4/6 inhibition with endocrine treatment. And they also tried to define the effect of pre-treatment RB1 health loss on acquisition of post-CDK4/6 RB1 loss of function variants. It was seen that RB1 health loss and other allelic imbalance have shorter PFS to CDK4/6 inhibition. And there is something called fraction genome altered, that is FGA, which is a measure of overall copy number burden. And if the high FGA is a poor prognostic sign, and it is enriched in other allelic imbalance. And it was seen that RB1 head loss is associated with shorter PFS that was independent of the FGA. And head loss do not confer short PFS if the patient starts on aromatase inhibitors alone. And head loss is not prognostic of any oral survival also. So, in summary, if you have RB1 head loss, it is associated with short PFS on first-line CDK4/6 inhibition plus endocrine treatment, but that is independent of the genome-wide copy number aberration. And RB1 head loss is not associated with short PFS if the patient starts on monotherapy with aromatase inhibitors. And it's not prognostic of overall survival and may be predictive of poor outcome of CDK4/6 inhibition rather than it's a prognostic biomarker. In conclusion, allele-specific copy number analysis allows for the discovery of clinically relevant genomic features which may load conventional somatic sequencing methods. And RB1 head loss may be predictive of PFS on CDK4/6 inhibition plus endocrine treatment and predictive of acquisition of RB1 loss of function variant as resistant mechanism. And they also tried to come with expansion of double hit hypothesis, that is development of second hit tumor suppressor under selective pressure of targeted therapy. Coming to the abstract 4, ESR1 F404 mutations and acute resistance to fulvestrant in plasma match study.
1 minute.
Yeah. Plasma match cohort A investigated the activity of fulvestrant in patients with activating ESR1 mutation ctDNA. Here present the baseline and end of treatment ctDNA to identify the potential resistant mutation to fulvestrant. Paired baseline and end of treatment plasma samples from patient enrolled in the plasma match study underwent ctDNA sequencing to identify the acquired mutations. For F404 analysis, MCF cells were transiently transfected with the estrogen receptor expression construct which contains wild-type ESR1 or a single ESR1 mutation or a compound mutation. And these transfected cells were treated with or without fulvestrant, and ERE luciferase activity was compared. And coming to the results, of 84 patients in cohort A, 69 had paired baseline and UAT sequencing.
So just give the take-home message.
Yeah. Yeah, conclusion, take-home message is this study identified a novel resistant mechanism to fulvestrant with F04 mutations occurred in patients with the pre-existing ESR1 mutation. And this F04 converts the fulvestrant resistance through loss of pi stack bounding, and it likely reduced the fulvestrant binding capacity, and this could be an identifying potential target to overcome endocrine resistance in future. Thank you.